Welcome. It's Friday. I've got to say that every Friday. You know, I always like to just, it's one way you get people happy. We say it's Friday, right? Cancel due to happiness. We don't want to cancel Friday, but you want to cancel the class is what you're talking about. Yeah. Oh, cancel the midterm. Oh, okay, yeah. I'm sure that'd be real popular then, yeah. Okay, let me give you a couple logistical things relative to the midterm, okay? So, first of all, we have a limited amount of time in which to give this exam, right? So, one of the things that you'll see is I'm very picky that nobody gets longer than 50 minutes, 50. So, I'll put a clock on the screen. You'll be able to see the screen. But when I say stop, okay, you stop, all right? That means if you're writing anything after I say stop, you're going to lose points. And my reason for doing that isn't to be mean, but rather to give everybody an equal amount of time. Now, I started that because a few years ago, in turning in exams in this class, sometimes there's a very long line doing it. I found people at the end of the line were getting an extra 5 minutes doing an exam, writing answers while people were turning things in. So because of that, I stop it right at the mark. And I will say stop. So when I say stop, if you're writing anything, even your name, you're going to lose points, okay? So when you get your exam, put your name on there first thing. Put your ID number on there first thing, okay? That way you won't have these issues at the end. Now, because we have a limited amount of time, I need to ask you to be careful how you sit. And now this is a big class and it's a big room. We can sit for part of the room in every other seat, and other parts we can't. So here's what the plan is going to be. The first 10 rows in the front, you can sit anywhere, okay? After the first 10 rows and all of the rows up there, all right, we're going to sit every other one. All right? Now, here's how it will work. You're going to line up every other one starting with this seat on this end right here being number 1, number 2, number 3. So you're going to be the odd-numbered seats. 1, 3, 5, 7. Okay? So the number 1 will start right here. The number 1 for this one will start right here. Okay? Now, if you come in and you see somebody sitting in number 2, don't move over to number 4 because I'm going to move them out of 2 into 3. Understand? If you have to have a left-handed seat or whatever, then my recommendation is get here early and do it. I'm left-handed. We didn't even have left-handed seats in the old days. I know you can do it if you don't have the right-handed seat. Okay? But if that's essential to you, get here early, get a seat that's there. But I will not let you change your seat so you can have a handed seat the way that you want. 1, 3, 5, 7, 9, etc. Okay? So count. Don't just move 2 over from that person there because they may be sitting in an even-numbered seat, and then I've got to move everybody over. Okay? So make sure you count things appropriately. For this section over here, and this is true above as well, this will be number 1 right here. 1, 3, 5, 7. Are we clear? So the first 10 rows, closest to the front, you can sit anywhere, but after the first 10 rows, you're going to be sitting every other seat. Okay? Makes sense? I will be videotaping you taking the exam. Yeah. That helps wandering eyes a lot because I've got a great camera. Okay? Now, you will also be turning in your exam, and I'll be videotaping that. So we won't check your ID, but we want to see your shining face turning in that exam. Okay? Getting out of here usually takes some effort. So I want you to be quick, get your exam dropped off, and get out of the way so other people can get their exam in because there's a class after us, and we can't run into their time. Understand? Okay, so be very careful with that. That's important. The TAs asked me to tell you that I will be answering all questions. Okay? And that's so you get consistent answers. That means I won't have to go jogging that morning before I come here because, believe me, going up and down and back and forth and in and out, I will be getting my exercise. Okay? So if you have your hand up, the TAs will make me aware of it. Just wait, and I promise I'll get to you. But make sure that you get your hand up. Don't come see me. I will come see you. All right? So I will come to where you are. Don't climb over people to get there. I'll be the one climbing over people. And even if you're in the front row, don't come see me. Just sit there, and I'll come talk to you. Okay? It works pretty well, you will have 50 minutes. The exam is the format that you've seen in the practice exam. And as I've said earlier, that this is an exam where time is sometimes an issue. I try on this exam especially to reduce that as an impact, but I find some people are more comfortable with calculations than others are. So you'll see some people leaving really early, and you'll see other people that will be here till the very end. And that's fine, okay? But the point is, budget your time accordingly. Don't put all your time into one question, and then you realize you've got a whole big blank section of the exam because you didn't pay attention to the time. The time will be shown on the screen, and you'll be set for that. Okay? All right, the other thing that people have been asking me about is review session. What's the deal on the review session? So I have, in fact, scheduled a review session. It is tonight. Well, it's late afternoon today. It starts at 4:30 today. It will go till no later than 6. We have to give the room up by 6, and it'll be right here in this room. And yes, I will videotape it. Okay? So I will get that posted as quickly as I can, and you'll be set for that. Okay. Any questions, comments? Clear as mud? Question?
So how, like, in terms of quickness, will it be posted by the weekend, the video of this session?
So I will try to get it posted. The rendering takes a while, the uploading takes a while, and technical things take a while. So I would certainly hope it'll be posted by early Saturday, but I will also tell people that every video I do, there's always the risk I'm not going to get it. Technical problems occasionally arise. So they're rare, but I can't promise anything, but I can promise to get it as quickly as I can, and my aim would be to have that up and available by Saturday morning. Yes? Yeah? Well, there are typically about 13 questions that are short answer. There are typically 2 to 3 longer problem solving, and then there's anywhere from 2 to 4 longer, not problem solving, but longer answer type problems. Okay? So that gives you a ballpark idea of where you are. Okay? Yes? You will not need a calculator, and you will not be allowed to use a calculator. Do not bring a calculator. Okay? Do not bring a calculator. You will not need one. You will not be able to use one. Okay? So it's designed so that you won't need to punch any numbers. Trust me. Yes? Okay, are you going to answer the questions on a separate piece of paper or on the space? You're going to answer right on the exam. Right on the exam. Okay? Okay? Everybody, yeah?
For anything like a graph interpretation, do you recommend that we bring a little ruler for anything?
For anything like a graph interpretation, do I recommend that you bring a ruler? The answer is no. You won't need a ruler or anything like that. Okay? If you need to do any drawing, I trust that you'll draw reasonably well. But what just happened there? Oh, okay. Hello. There we go. Okay. Other questions? Are we set? Okay. Oh, yeah. So the material ended at the end of the enzyme lecture. Yeah. Okay. All right. Well, let's move forward. We're going to talk today about, we're going to flesh out a little bit more the catalytic strategies that we started talking about last time. Okay. So, as I said, we learned something about enzyme structure and some general things about function and kinetics. But now we want to really get in up close and personal and learn something about how enzymes actually function. All right. So, as I said last time, we use serine proteases as our model enzyme because they're very well studied, and there are a lot of them. And as you will see as we go through these enzymes, that you're going to see some common themes that are arising that will help you to understand the actions of many enzymes. Not all enzymes, but many enzymes. Okay. So, I'll remind you last time I talked about the catalytic triad, and this is the place where I stopped last time. All right? So the catalytic triad is at the active site of chymotrypsin. We're talking about chymotrypsin here, all right? And I'll remind you that what was happening here was the whole process started with the proper binding of the proper substrate. What does that mean? Well, when we talked about the specificities of the proteases, we saw, for example, trypsin cut next to lysine and arginine, all right? It doesn't cut next to aspartic acid. So if this enzyme were to bind an aspartic acid residue in a protease, it wouldn't have the right binding site, it wouldn't bind properly, and the enzyme would not go through the steps that I'm talking about here. Only if the enzyme bound the proper substrate will these following reactions occur. Now that's important to understand because it really tells us what specificity is and what specificity means. Specificity means only certain things that the enzyme is going to work on. And that is determined by the things that the enzyme will bind. And the binding of those things causes changes that result in a reaction. So if the wrong thing binds, these changes don't happen, a reaction doesn't occur. The right thing binds, the changes happen, a reaction does occur. Okay, so just briefly reminding you what we did last time was we said, okay, we've got this chymotrypsin which binds a substrate, it binds an amino acid within a protein that it recognizes, and it knows it has to cut on the carboxyl side of that. Okay? That binding causes a very slight change in the shape of the enzyme. You saw how hemoglobin's very slight change in shape caused it to have the phenomenon known as cooperativity. This slight shape change, I always have a hard time saying that, this slight shape change, okay, causes the enzyme to catalyze a reaction. All right, so what happens? Well, what happens is in the process of this slight shape change, the aspartic acid, as I noted, is moved closer to the histidine, closer than it was before. Because it's closer and because it's negatively charged, it changes the electronic environment of the histidine itself. I like to think of it in simple terms as saying that it makes those electrons in the histidine move to the right. Okay? That makes the right side of histidine more negative. That makes that proton very attractive to that negative charge, and the proton, as a result, gets taken off. Now, the taking off of that proton is essential for the catalytic action of serine proteases. It's essential for the catalytic action of every serine protease. This mechanism I'm talking about here works for all the serine proteases, and there are many. The result of taking off that proton causes formation of an alkoxide ion. The alkoxide ion is a negatively charged oxygen, as you see there, and it's extraordinarily reactive. Okay? Now, that's what happened to get us to about this point right here. Here, schematically, is the protein that's being cleaved. You see there's one end called R2. There's another end called R1. This protein has bound properly into the active site. We'll see later the active site has a special pocket called the S1 pocket that it physically binds into. Yes? I'm sorry? The active site of the enzyme. Okay? So the active site of an enzyme is the place where the reaction is being catalyzed. So we're looking right up close and personal where these reactions are occurring inside the bowels of this enzyme. Okay? All right. So we've got everything going on here. Now, this alkoxide ion, which isn't even shown on this figure, this alkoxide ion is very reactive, and it acts as a nucleophile. Anybody remember what a nucleophile is? No? I see a few yeses. Good, good. Okay. Nucleophiles, well, nucleo meaning nucleus, phile meaning like. This likes nuclei. All right? It's electron rich. That oxygen is negatively charged. It really likes that carbonyl carbon in the peptide bond. The carbon is electron poor because the double bonded oxygen is taking electrons away from it. So you've got an electron-rich oxygen on one side, an electron-poor carbon on the other. They make a nice natural match. So the alkoxide ion attacks that carbon. It forms a covalent bond with it, and in the process of forming that covalent bond, the peptide bond is broken. So the alkoxide ion attacks the carbonyl group. The peptide bond is broken as a consequence of that. And there's a peptide bond now, a covalent bond, that's holding part of the protein to the enzyme. Okay? Actually, it's this part over here. It's holding part of the protein to the enzyme. Here's where the oxygen attacked that carbon. Now there's a covalent bond between the two. Here's where there was a peptide bond, but you see the peptide bond has been broken. And the only thing that's holding the rest of the protein in place is a hydrogen bond. We all know hydrogen bonds are not as strong as covalent bonds. So this other part of the peptide that got cut off is now free to fly away. And it does. So when I talked in class last time about a fast step of catalysis in serine proteases and a slow step of catalysis, you've just seen the fast step. You've just seen the fast step. The formation of the alkoxide ion, the attack of the carbonyl group, the breaking of the peptide bond, and the release of one of the peptides. Bang. That happens very quickly. Now we enter the slow phase. The slow phase has to release this other peptide from this covalent bond where it's attached to that oxygen that was part of serine. Okay? So now we've got to release this. Because if we don't release this, as I said last time, we just made a suicide enzyme. It could only react once. If this doesn't get released, this enzyme is not going to work anymore. Enzymes, if they're going to continue to function, have to release this transient bond that we have right here. Okay, so the next steps that we have in process basically allow this to happen. Okay? So we have step 1, step 2, step 3. All right, so we see up here, we see this guy here has now left the enzyme, and we're left with this guy behind. Well, how do we break that bond between the other peptide and that oxygen? It turns out it's a very similar process. It's just not as fast. All right? Water has to make its way into the active site. Okay? Water plays a role in the next steps in releasing this peptide. Water comes in, and water looks like this, HOH. And there's that electron-rich histidine. Okay? That electron-rich histidine is capable of doing what? All right? It is capable of pulling off a proton. You saw that earlier. This electron-rich histidine now pulls off a proton, and water has a hydroxide that's negatively charged. Guess what? That negatively charged hydroxide is a nucleophile. It attacks the same carbon. This time when it attacks the same carbon, what happens is the bond that's broken is between the oxygen and the carbon, and the second peptide gets released. Now what we've done is we've basically gone through and gotten back to the starting point, which was over here. We've broken the peptide bond, we've released one fragment, we've released the other fragment, and now we've regenerated our original enzyme. Okay? Everybody looks stunned to silence here. Questions? Yes, sir? Oh, okay. Go to an auction, you put your hand up like that, you bid. You just bought my valuable baseball card collection for a very high price. Okay. Makes sense? Wow. How much of this do we have to know? That's what I always hear. Are we going to have to draw this on the next exam? Hope not. Well, as you know, I think that regurgitating things like that really aren't necessarily the best ways to learn things. But there are some important steps in this process that I think anybody should know. And there's a couple things, at least one thing I haven't told you that I'll tell you a little bit more about in a minute. Okay? Let's think about the steps. Let's think not about the structures. Let's think about the steps. What's happened in this process? The proper substrate bound. Number one. Right? The binding of the proper substrate changed the shape of the enzyme, which caused an alkoxide ion to form in the catalytic triad. The alkoxide ion attacked the carbonyl carbon of the peptide bond and broke the peptide bond. Okay? One fragment got released. The other fragment had to be released by water. That's basically what happened. Right? That's not complicated, I hope. Yes, question? Oh. I keep looking for questions. Sorry. Okay, so couple things, a couple structural considerations here. One is I talked about the S1 pocket. The S1 pocket I'll show you a figure of in a minute that I hopefully will make it a little bit more clear to you. The S1 pocket is part of the active site. It's a specific little pocket, we can think of it like a little depression, where the side chain of an amino acid can fit. This is how specificity is determined. The right side chain binds in this pocket, then the enzyme goes through these shape changes and the catalysis occurs. S1 pocket gives the enzyme specificity. Okay? Another thing I didn't talk about was actually a structure that you see here, and in fact it's labeled up here, the oxyanion hole. The oxyanion hole sounds kind of complicated, and it actually is more complicated than I'm going to tell you here. But suffice it to say that the oxyanion hole is there to stabilize a very unstable intermediate. It's there to stabilize a very unstable intermediate. That unstable intermediate is right here. You'll notice that right here, the peptide bond hasn't yet gotten broken. What you want this unstable intermediate to do is to break bond in the right place. And it turns out that because the oxyanion hole is there, it keeps this unstable intermediate from reacting with the enzyme. If it reacts with the enzyme, nothing more is going to happen. The enzyme is dead. It stabilizes this guy enough so that the bond breaks in the place where it's desired. It doesn't react with the enzyme, it reacts with itself. The substrate reacts with itself, and this bond is broken, causing the release of the fragment. So the oxyanion hole plays an important role. It plays it down here as well. This intermediate is unstable, and instead of interacting and reacting with the enzyme, the bond now between the carbon and the oxygen is allowed to break. And then the second peptide is released. Okay. Where's the oxyanion hole? Well, if you look at this, you realize it's got to be at the active site too. All these things are playing roles all in the same region, same section of the enzyme. Okay? Makes sense? I understand you guys be so quiet. Questions? Everybody's thinking about the exam. I'm not paying any attention to him. I'm studying. Yes?
In step 6, is there a proton transfer that's not shown between the oxygen and nitrogen?
On step 6, is there a proton transfer that's not shown between the oxygen and nitrogen? You're talking about, I'm not sure where you're talking about.
Off of the serine and off of the histidine?
So there's a proton transfer that's happened actually from here to here. What I didn't show you was a proton coming off of this guy up here. Okay, but it does come off, as you see, when this guy leaves up here. But I'm not sure where you're saying here.
Next step, that the oxygen is connected to hydrogen instead of the nitrogen.
The oxygen is connected to hydrogen instead of the nitrogen. Here?
Yeah.
So this, okay, so I see what you're saying. You're asking, is it going from the nitrogen back over to the oxygen? Yes, I'm sorry. Yeah, that's the case. So this is released back over in this direction because this oxygen, again, is going to be reactive and it's going to be very electron rich. So now we're releasing this. The enzyme is going back to its original shape, and this hydrogen is released back to the oxygen. I'm sorry. Yes, I didn't understand what you're saying, but that's important because we need to regenerate the OH in order to regenerate the enzyme. Right. So the beauty of this is we go through all these steps and we end up right back where we started, except for the protein has been broken into 2 pieces. That's cool. Yes?
Can you repeat the first step that you said? You said the first step is something with the oxide attached carbonyl and peptide bond.
Okay. Well, the first step is the proper binding of the substrate. Binding of the proper substrate into the S1 pocket causes a conformational change in the enzyme that changes the geometry of that catalytic triad. It's that change in geometry that causes the alkoxide ion to form and then the other steps to happen. I think that's what you're asking about, right? Okay. Yes?
How much faster is the fast step?
Yeah, that's a good question. I couldn't tell you, but you saw on the figure that there is a difference. I would guess you're probably talking about 3 or 4-fold. Okay. Let's see. Let's say a little bit more about this. So individual steps there. I'm not going to go through all those. Here's the oxyanion hole, which is there. And no, you're not going to draw that structure. Just showing you that there is actually a little pocket and a little place to help stabilize that unstable intermediate that's in there. Okay. I won't go through that. Okay. Now, I told you that serine proteases are very similar to each other in function, and I told you that they all had a catalytic triad. But this next figure, I think, is very good because it shows us 2 different serine proteases. One is trypsin, and one is chymotrypsin. You saw that the specificities that those 2 enzymes had were somewhat different. Chymotrypsin tends to work on aromatic and nonpolar type side chains, whereas trypsin tends to work on lysine and arginine. Okay? But when we look at them structurally, one's in blue and one's in red, we don't see a very big difference between them. So that reminds us of a couple of things. We've talked since day one how structure is essential for function. These are both proteases. These both have a catalytic triad. We might imagine that getting the orientation of that catalytic triad is going to be pretty important, so it's not too surprising that the surrounding structure that enables the catalytic triad to form would be similar. And in fact, that's exactly what we see. When we compare these 2 enzymes, we see the primary differences in structure arise from their S1 pockets. S1 pockets. Or they're on the outside. Things on the outside don't have as much impact as things in and near the active site. Okay? All right. Now, let's go back and think about S1 pockets. S1 pockets are pockets, like I said. They're a place where a side chain of an amino acid from a protein can bind. This shows the chymotrypsin, this shows trypsin, and this shows another one called elastase. All right? Trypsin, you remember, recognizes and cuts next to lysines and arginines. Those have side chains at physiological pH that are positively charged. Look what's at the bottom of their S1 pocket. How convenient. Right? Positives attract negatives. This is really nice. Chymotrypsin tends to work on things that are fairly nonpolar. Do you see any charges in there? Mm-mm. Okay? Elastase tends to cut fairly nonpolar things, but with small side chains. Look, they physically block access to the bottom of it so that you can't squeeze in an aromatic ring into there. The S1 pockets have configurations that allow us to have specificity. If I had an enzyme that cut next to only aspartic acids and glutamic acids, what do you suppose would be at the bottom of its S1 pocket? Positive charge. You might be very expecting to see lysines or arginines down there, right? So this principle is very simple, but it's very, very important to understand. So the S1 pocket gives that specificity that is important for the enzymes to work on. Okay. Now, this figure may look a little confusing at first, but it's kind of cool. Right? And what this figure shows us is that the catalytic triad is really important. All right? So you should never take my word for something. I've been telling you the catalytic triad is important, but the way in science that we determine something is important is by experimentally verifying it. Okay? So trust isn't something that is a way of finding things, right? So somebody said, well, I want to make sure it's important, right? So they take chymotrypsin, or trypsin, doesn't matter, it's a serine protease, and they measure its activity. Okay? So what's KCAT tell us? This is on the next exam, guys. What's that? It tells us basically how rapidly the enzyme is turning over substrate and making product, right? How rapidly it's making product. Okay? So it's a measure of the speed of the enzyme. All right? Here's the speed of the enzyme that's got a perfect catalytic triad. What if we mutate the serine to an alanine? what happens? The activity of that enzyme that has an alanine in place of serine goes down by 1, 2, 3, 4, 5, about 6 orders of magnitude. 6 orders of magnitude corresponds to 1 million. This guy is 1/millionth as active as this one is. All right? If we mutate the histidine to an alanine, we see it's also about 6 orders of magnitude, about 1/1,000,000th as active as the wild type. It tells us that the serine is really important. It tells us that the histidine is really important. If we take the aspartic acid and we mutate it to an alanine, we see about 1, 2, 3, 4 orders of magnitude, or about 10,000. It's still important, but it's not as important as these two are in the overall process. Interestingly, if we take and we mutate all 3 of them, we see an activity that's roughly the same as if we mutated either the serine or the histidine, telling us that these are critical residues for function. If we have a completely uncatalyzed reaction, we see something down here, which tells us that even here we have a little bit of activity. Okay, so these are critical for the function of the enzyme. Now you don't have to trust me. You can say you've had evidence shown in front of you. Yes?
They tried it, I mean, they mutated them all to alanine, but have they tried it changing it to something that's a more similar?
Our question was if we change it to, say, from aspartic acid to a glutamic acid, okay, what would we expect that would happen? That might be a good exam question, but now you know what to study for. No, so it's a good question. So let's say I mutated that aspartic acid to a glutamic acid. What do you suppose would happen? What would be your prediction? In between, okay. So you think it would be fairly active or not fairly active? Fairly active, okay. What might stop it from being fairly active? What's that? Well, substrate's going to be the same no matter what you do. So, right. So what might stop it from being very active? You would predict it would probably be fairly active because glutamic acid is going to have a side chain very much like aspartic acid, but it's not going to be identical. And so you might have a geometrical or a configurational impediment that might stop it from being active at all. So you don't know until you do the experiment. Yeah, but it's a good question. Okay. You guys are a little dead. What do you say we do a song? Okay. To reinforce what we just talked about. This is an easy one to sing. It's called the New Serine Protease Song. It's to the tune of Rudolph the Red-Nosed Reindeer. Okay. All serine proteases work almost identically. I can't hear you. Using amino acid triads catalytically. First they bind peptide substrates, holding on to them so tight, changing their structure when they get them in the S1 site. Then there are electron shifts at the active site. Serine gives up its proton as the reaction goes on. Next, the alkoxide ion, being so electron rich, grabs peptide's carbonyl group, breaks its bond without a hitch. So one piece is bound to it, the other gets set free. Water has to act next to let the final fragment loose. Then it's back where it started, waiting for a peptide chain that it can bind itself to. Go and start all over again. All right. What do you guys we say? Let's stretch. Get up and stretch. You're really a dull group today. Stretch. Stand up, stretch, take some breath. We've still got more to do. Huh?
Carbonic anhydrase will still be there for you.
Yes, it'll still be there for you. Come on, jumping jacks, guys. Come on. Right. Ah. All right. Okay. Now, as I said earlier, the principles that you learn with serine proteases extend to other enzymes. other classes of proteases, and even things that aren't proteases. Okay, so some of the things we learn here can be very valuable for the next steps. All right, here is an enzyme known as papain, and papain is an enzyme that we call a cysteine protease. A cysteine protease. Well, a cysteine protease, as its name suggests, has a cysteine that plays a role in the active site. And I'd like you to think for a moment, cysteine has a side chain of SH, that's the sulfhydryl, and the serine proteases had a side chain of OH. What do you suppose is going to be the nucleophile here? S minus. If we look in the geometry of what's there, we see, hey, right next door, there's a histidine, just like we had with the serine protease. Now, we don't have an aspartic acid here. We don't have a catalytic triad, but you also saw that the aspartic acid wasn't quite as critical as the other two were. This guy grabs a proton off of cysteine and makes an S minus, and the S minus attacks the carbonyl carbon, and we see exactly what we saw going through the serine proteases. Okay? Cysteine proteases are very easy to understand in comparison to serine proteases. All right? Here's an enzyme called an aspartyl protease. I'm going to show you the mechanism for it in a second. But it's called an aspartyl protease because it has two aspartic acids that are positioned in an appropriate place in the middle of the active site. Okay? I'll say something about that mechanism in a second. And metalloproteases are the last group of proteases I'll mention, and they use a zinc in the process of their catalytic action. Okay, well, here's the cysteine proteases. As you saw before, exactly the same thing happens. Here's the pulling of the proton by the histidine, formation of the S minus, attack of the carbonyl group, etc. We see this exactly like the serine protease. When we look at the aspartyl proteases, we see something different that's going on. Okay? Something different's going on. But you'll see similarities to the other. All right? Now, here's an aspartic acid residue. Here's an aspartic acid residue. We're up close and personal inside the active site. We have the substrate, the proper substrate that is bound. And these aspartic acids have gotten into position to start the process. Okay? What happens in the positioning of these guys together is that this negatively charged oxygen right here is brought into close proximity with water again. And you saw how that negatively charged oxygen can pull a hydrogen off of water. And what happened when we did that inside of the serine protease? The hydroxyl became reactive, it was a nucleophile, and it attacked the carbonyl carbon. We pull this hydrogen off of this water. We make a reactive hydroxyl, which is a nucleophile which attacks the carbonyl carbon. We see the same thing going on. Okay? Now, there's something that's different here, though, than the serine proteases. Okay? It turns out that this enzyme doesn't have a fast step and a slow step. My question to you is why? Yes? So what she's saying is that this oxygen is going to bind up here, this hydrogen goes down here, and you basically release everything. Is that what you're saying?
Well, you end up kind of flip-flopping which one has no H or something.
Okay, not quite, not quite. There's a more fundamental reason why there's not a slow step in this process. Yes?
One step.
It's one step. Okay? In the other one, remember when we had that serine protease, we had the alkoxide ion, and we formed a covalent bond between the serine that was part of the enzyme, right? The serine was part of the enzyme, so when we made that covalent bond, we had to release it. Here, the attacking nucleophile is not part of the enzyme. The attacking nucleophile is coming from water. So when it attacks, hydroxide goes one place, hydrogen goes another place, sort of like you said, okay? The thing breaks and everything falls apart. One step, okay? It's not a two-step process. Okay, very good. Here is a metalloprotease. Okay? Metalloprotease is going to look to you like an aspartyl protease because, well, here's a zinc that's being held in place in the active site of the enzyme. Here's a water that has come in. The zinc helps to hold the water in place by organizing the oxygen. The oxygen is relatively negatively charged. The zinc is positively charged. So the zinc is helping to position this water so that something up here, and this varies from enzyme to enzyme, so that something up here can take away the hydrogen. Takes away the hydrogen, what do you have left? A reactive hydroxide. That reactive hydroxide acts as a nucleophile, and you guys know the rest of the story. One step or two steps? Going to have a fast step and a slow step here or not? You're not going to have a slow step. Because again, the attacking nucleophile is not a part of the enzyme. Okay, very good. Catch our breath here for a second. Let's go to another group of enzymes. One we've been talking about already that hopefully you will see some similarity here. Carbonic anhydrase is, I've tried to convince everybody, a magical enzyme. All right? You remember that carbonic anhydrase had this phenomenal kcat. It had a phenomenal kcat. All right? It could make 1 million molecules of product per molecule of enzyme per second. Here's the reaction that carbonic anhydrase catalyzes. All right? It catalyzes the combination of carbon dioxide with water to form carbonic acid, which will ionize to form carbonate, bicarbonate. Okay? That's the reaction it catalyzes. It's extremely rapid. Okay? Let me show you how that process occurs. Well, before I do that, let me show you some interesting things about the enzyme. Kcat is a constant for an enzyme under a given set of buffers, right? Well, if we change the pH of the buffer, not surprising, we're going to change the kcat, because if we took and we denature the enzyme, what's going to happen to the enzyme? No kcat, right? If we take this enzyme and we measure its kcat at a very low pH, relatively about 5.9, and we keep increasing the pH and we measure the kcat, we discover something interesting. We discover that the kcat for this enzyme maximizes up here at about 9. Okay? So when you saw the book showing different kcats, they were actually measured at different pHs. At physiological pH, this guy's about 600,000 to 700,000. We don't get 9 we don't get 1 million unless we go up to a pH of 9. Well, that's kind of odd. Why do we have an enzyme that has a maximum activity at pH 9 where our body isn't having a pH like that? All right. Well, when we look at the mechanism, we can discover what's happening. All right. This enzyme uses a mechanism like we saw with the metalloproteases. There's a zinc, there's a water. If we look at what's happening, we discover this. We see the binding of the zinc in the water. We see the loss of the proton and making a hydroxide. We see the hydroxide attacking a carbonyl carbon, and we see the formation, ultimately, of bicarbonate. So why does this have such a high pH that's optimal at? The reason it has such a high pH is what happens at a higher pH? Is water more likely to lose protons at a low pH or at a high pH? At a high pH, right? This loss of a proton that occurs here is much more likely to occur in a pH 9 solution than it is to occur in a pH 5 solution. This turns out to be a limiting step in the process. By having this at a higher pH, we're favoring this reaction, and once we get to this reaction, everything else follows quite readily. The enzyme really likes pH 9. For some reason, the enzyme doesn't like pH 12. Do you know why? Yes, you do. What happens to it at pH 12? It's probably going to denature, right? You got it, okay? So, even though we may favor conditions in the active site, we're not favoring the overall structure of the enzyme. So we've got limits. We have limits within which we have to work for enzymes to be functional. Make sense? So if the carbonic anhydrase is in a lower pH, it will be less active and it will be less able to make carbonic acid. Now think about this. What does that mean? What's carbonic acid going to do? Carbonic acid is going to release a proton, right? And thereby make it even more acid, right? And we're not going to tolerate that acid too much. So having this sort of built-in control, keeping from making too much carbonic acid, which would keep us from making even more protons, actually can play a role, right? Acidifying the blood is not something that you want to do too much. Yeah? Is that why you do what?
Give bicarbonate sometimes coating.
So bicarbonate itself will act as a buffer. So giving bicarbonate actually can help with that process, yes. Yep. Okay. I think we've done enough. What do you guys say? Okay. Let's